Hi everyone, my name is Leonid and today we are going to learn about peptides analytical features and application designed at Datagrok. This application aims to deal with peptide sequences and associated activities to process sequence activity analysis. From the computational and analytical point of view, peptides are considered as an intermediate case between small molecules and proteins, and thus require different combination of analytical methods used for both of these classes in adequate proportions. We performed the review of existing solutions and methods being in development to enhance the Datagrok platform capabilities to cover the vast common cases, though the extensions are also on their way. In this demonstration, we operate with a column of aligned amino acid sequences, namely peptides and corresponding half inhibitory concentration values or IC50. You don't need to run a special application to process peptide string, as Datagrok automatically detects them and renders in a way of highlighting hydrophobic and hydrophilic regions in the sequences and regenerating structures. Here we are going to implement a simple sequence activity analysis to find the most potent pairs of amino acid residuals and positions in which these residuals are located. To do this, we open the property panel by clicking the column header, and in this panel we choose the preferred activity column and possible data transformations. The most active sequences now have the lowest IC50 values, and the distribution of IC50 tends to be biased, as could be seen in the histogram. We should now choose -log transformation to get PIC50 values, and then launch sequence activity relationship analysis. When the analysis is launched, we can check the transformation for analyzed values, and now they seem to be distributed normally. The analysis itself provides a bunch of tables for the sequences. To facilitate the interpretation of analysis results, the sequences are now replaced by alignment table, where the splitted sequences could be found and where we can inspect each residual in each position and compare it to composition analysis plot at the top of this table. To visualize the entire dataset and filtering results, in the left bottom we show the peptide space, where entries are mapped using Levenshtein distances. The results of the analysis are presented as a table with residuals in rows and positions in columns. Here the positions versus monomers could be investigated to yield inactivity difference. Size of the points in the table show the mean difference in activity values for a subset having specified residual in a specified position and the whole remaining dataset. Color of the point responds to significance levels of such difference. For example, now we choose the 14th position with lysine. The dataset has 205 entries with this case, and this subset will have 0.8 log units activity difference in average with all other entries. The p-value indicates the significance of this difference. One may estimate these differences using and comparing the densities in the property panel. In the right bottom table, the possibly most potent sequence is generated using the calculated mean differences and selecting the highest and the most significant ones for each position. So thank you for your attention. The additional information on peptide sequences analysis with the platform could be found on Datagrok wiki.